Genetically engineered microbes have long been used as living foundries for the production of drugs and fine chemicals. More recently, different laboratories have combined bacteria with semiconductor nanotechnology. When you put semiconductor nanoparticles on the surface of the microbes, they function similarly to solar panels on the surface of a house. So, in other words, they're able to convert light energy into other forms of energy to boost the biosynthetic potential of the cells. The first biological inorganic hybrids, or biohybrids, focused on organisms that could convert carbon dioxide into relatively simple organic molecules that could be, for example, used as alternative energy sources. In a project led by two postdoctoral fellows in my lab, Junling Guo and Miguel Suastegui, who come from vastly different disciplinary backgrounds, we've extended this concept of biohybrids to baker's yeast, which is already an industrial workhorse and can be genetically manipulated relatively easily to produce a more complex range of molecules. Genetically programmed yeasts are broadly used in biomanufacturing to produce high-value chemicals and medicines. A yeast cell is like a tiny factory with a highly complex system of metabolic pathways, many of which are linked to each other. Engineered yeasts use renewable carbon sources to generate energy and precursors for the synthesis of chemical products, some of which are very useful for pharmaceutical and other industries. For example, the yeast metabolite shikimic acid is a key precursor to the influenza drug Tamiflu. The pathway that produces shikimic acid starts from the raw material glucose, which is taken up as a carbon source into the cell, converted over several steps into 3-dehydroshikimic acid, or DHS, and eventually to shikimic acid. The final conversion of DHS to shikimic acid consumes the energy stored in the molecule NADPH. The energy-depleted form, NADP+, is recharged by the pentose phosphate pathway and becomes NADPH again. However, the pentose phosphate pathway consumes glucose and diverts some of the yeast's carbon flow away from shikimic acid production. To enable yeast cells to funnel more of the carbon atoms from glucose towards shikimic acid production, we have genetically modified them to shut down their pentose phosphate pathway. However, this also means that the energy carrier, NADPH, needed for shikimic acid synthesis is significantly reduced. To solve this energy crisis, our team now has enabled yeast cells to use an external light source as an alternative energy source that drives their metabolism. The system works by installing tiny solar panels in the form of inorganic nanoparticles on the yeast cell surface. The solar panel-like nanoparticles are made of a semiconductor material, indium phosphide, and coated with polyphenols, which function as a glue to attach the nanoparticles to the yeast cell surface. Once attached, the nanoparticles absorb light energy and convert it into electrons that they hand over to the yeast cells, which in turn use the electrons to convert NADP+ to NADPH in their cytosol, thus accumulating energy to fuel metabolic processes. In this way, the yeast cells are able to funnel more carbon atoms that they derive from glucose towards shikimic acid, significantly boosting its production. Combining genetically engineered yeast with an efficient semiconductor component provides biochemical energy to its metabolism without being toxic, which has been a problem with previous biohybrid systems. In this proof of concept study, we focused on shikimic acid as a product because its biosynthetic pathway is well understood and because it's an important synthetic precursor in the antiviral drug Tamiflu, other medicines, nutraceuticals, and several other fine chemicals. However, this approach also creates a scalable blueprint for future biohybrid technologies in which the identity of the semiconductor and the engineered cell can be varied in a plug-and-play fashion, greatly expanding the range of drugs or fine chemicals that can be produced in a cost-effective biomanufacturing process.